pitch at the undergraduate research symposium next week. So any feedback that you guys can give me on the poster or the presentation itself would be appreciated at the end. So to begin, this past year I studied Alzheimer's disease and used C. elegans as a model. And basically, let me zoom in. What Alzheimer's is, it's essentially a progressive neurodegenerative disease that causes a decline in mental and physical functions. And there's many characteristics, but generally speaking, it starts off with memory loss and challenges in problem solving and planning, and then eventually leads to physical loss as well, like the inability to like talk and eat, walk, etc. And in the brain, it's characterized by a physical change. And essentially what happens is that there's a protein buildup and aggregation of two proteins specifically, the amyloid beta protein, which was the focus of my research, and the tau protein. And amyloid beta forms these plaques in the synapse as well as around the neuron itself. And the tau protein coils around the neurons. And no one exactly knows why this happens, but there's three major hypotheses. The first one is the amyloid cascade hypothesis, and that basically states that these two proteins are present in everyone, even in healthy people. But in Alzheimer's disease, essentially these proteins build up over time, and as you get older, they don't dissolve back in. And it eventually later in life, it reaches a toxic level. The second hypothesis is because neuroinflammation and oxidative stress in neurons is also characteristics of Alzheimer's, but these are also characteristics of an infection anywhere in your body. So the second hypothesis is that Alzheimer's is caused by a virus or a bacteria, and there are several being studied. The most common one is P. gingivalis, which is the same bacteria that causes gingivitis in your mouth. And basically, these two proteins in the brain are produced as an antimicrobial effect, and it eventually leads to Alzheimer's. And the third hypothesis is familial Alzheimer's disease. And basically, this happens when there is a mutation in the C99 fragment in your body, and that leads you to naturally overproduce actually the amyloid beta plaques, leading to Alzheimer's. So the goal of my project was to get amyloid beta produced in C. elegans. And basically, C. elegans is a very simple nematode. It's a worm that makes it ideal for research because it's transparent and it has a short lifespan. So you can monitor it easily over its growth. And it has similar biological and molecular pathways compared to humans. So whatever works on C. elegans can be applied to a certain degree in humans. So how exactly in humans is amyloid beta produced? So you have a protein in all of your cells called the APP, the amyloid precursor protein, and in the brain, it's cleaved by two enzymes. The first enzyme is the beta secretase enzyme, which cuts off the first half. What's left is called the C99 fragment. After that, the gamma secretase cuts off within the membrane, within the neuronal membrane, and it releases amyloid beta extracellularly, which then causes it to form plaques around the neurons. Worms, the C. elegans worm, has a similar protein called the APL protein, but because it doesn't have the amyloid beta fragment inside of it, like the human ones does, and worms also don't have a beta secretase, they have an alpha secretase. So naturally, they can't produce amyloid beta, and they can't exactly develop a disease similar to Alzheimer's in humans. So my goal of the project was because we had to get amyloid beta to be produced, my goal was to get the C99 protein to be produced initially in the worms. And then the hope is that the worm's natural gamma secretase will cleave the C99 protein, and amyloid beta would be produced extracellularly. So to get C99 produced, I started off with making a plasmid that would produce the C99 protein. And to do that, we used two plasmids that we merged together, essentially, through Gibson assembly. And basically what we did is that we took the GH8 plasmid, which has these three important genes highlighted right here. The green one and the blue one are necessary for all DNA editing in worms. They're recommended, basically. The mTERI gene is the gene that we took out of this plasmid through amplification, and then we added this orange C99 gene after Gibson assembly. So we got this final plasmid which had the two necessary the two necessary genes for gene editing in worms, and then we also had the C99 fragment inserted within it. From there, I sent this plasmid off to sequencing, and sequencing is basically a process that ensures that the fragment that you inserted into the plasmid was oriented correctly, and there's only one insertion, because errors can happen with the DNA ligase and polymerase, and sometimes you get 3 or 4 insertions. So once that was confirmed, microinjection can happen. And basically with microinjection is you take this plasmid and you add in another plasmid, like as you can see here. So you add your target plasmid as well as the marker plasmid. And the marker plasmid contains some sort of marker DNA that will allow you to distinguish between worms that you were able to successfully microinject and worms that you weren't able to successfully microinject. And how that happens is that when you insert a plasmid into the gonad of a worm, the DNA is cleaved and assembled into these large chromosomal arrays. And all of the progeny will get a random assortment, something like this, right? So the ones that get both the marker and the target will be transformed, and the ones that don't, you just have to throw away. So once we were able to transform the worms, the next step is to assure that amyloid beta is being expressed extracellularly. So how far I was able to get before the lab shutdown was, over the past year, I was able to basically confirm that we were able to amplify the C99 gene out initially. That was the first step. The GH8 gene and the GH8 plasmid took a very long time. This process took me the longest, and it ended up taking several months. I was able to eventually get it to work, but we had to, because it was such a huge plasmid that we were amplifying, we had to use different temperatures and extension times, number of cycles, and it was essentially very tedious and laborious. After eventually, when I did get that to work, we were able to ligate successfully, and when we sent off 20 samples to sequencing revealed that only one of them was successful in ligating properly. So the C99 was inserted and oriented properly. So before lab shutdown earlier in the semester, I was able to get microinjection to work as well. So if I zoom in, the marker DNA, by the way, that I used is called mCherry, which is basically, it's a protein. The mCherry gene produces a protein that fluoresces under light, and it targets the pharynx, which is like the neck area in the worms. If you zoom in, you can see some of the worms are basically lit up. So the hope is the worms that are being lit up also took up not only the marker plasmid, but also the C99 plasmid, the one that I'm hoping will produce the gene. And so from there, once lab reopens up again in the future, I'm hoping to go back and check the worms that fluorescence, hoping that they contain the C99 fragment as well, and then use real-time PCR to ensure that the amyloid beta is being produced. And then if that's being produced, then we can start imaging the worm's neurons over their lifespan and see how exactly amyloid beta causes neuronal degradation. And so I think that's about it. Any questions?
Mariam, that's really interesting. So, like my psych textbook talks about it all the time, how like when it talks about like causes of Alzheimer's, it just gives a list of things that you should know. And at the top is always like, we don't know why this is happening, but we know for whatever reason that beta amyloid plaques are like super common. So your research is really interesting, that you're like behind the scenes of like such a prevalent like disease and seeing like, I guess like on a molecular level, like what's going on. So that's really interesting. Shabazz, psychologist. Yeah, no, but you did a really good job talking about like the whole methods and everything too, because I feel your pain on like making sure that whatever, like amplifying a really big plasmid and making sure it's actually there, because the worst thing is spending months in a lab and then like getting your sequencing back and seeing that like nothing was successful.
Exactly. Yeah.
Like this, yeah, and I think it's really interesting what you did.
All right, thank you so much.
Miriam, I first want to say I'm very impressed with your research.
Thank you. Anything else? All right, then I can start.
I think I muted myself. But I do have a couple questions. Sorry.
Mm-hmm.
You said the humans secretase over here. It's a different. You said it's like a gamma secretase. Does the worms have the gamma secretase too, or is that different?
Yes, so that's my hope, that once I'm able to get the C99 fragment being expressed, the last step hopefully would be that the worm's natural gamma secretase will cut the amyloid beta and express it extracellularly.
Okay.
Because in the worms, they do have the similar protein that's also cleaved in two different places, the APL. But while they don't have a beta secretase, they do have the natural gamma secretase that's also in humans.
Okay. And then, hold on, I wrote down what I was going to say. Oh, since they don't naturally have this alpha beta fragment, are you concerned about some immune response that they might have? Like once it's produced and floating around, like that would be different from, like.
Naturally. Yeah, that's a possibility. But so the research that the grad student that I work with is conducting, he's actually infecting the worms themselves with P. gingivalis, because P. gingivalis causes the tau protein to be overproduced in human brains. And he hasn't exactly had any immune response from worms, other than the fact that sometimes they will just randomly die. That's the only problem that he's been having. These worms they're very, very simple animals. Like they only have a couple of organs. They don't really even respond to touch that much. So hopefully that's not a concern that I have to deal with when I do get to analyzing. But that was a good question. That's something to look out for, I guess.
Okay. And then the last question I had was, how did you amplify the plasmids? These are not done in eukaryotic cells, or like?
No, they were done through PCR. So
okay.
Yeah. So we use PCR to amplify the GHH, except for the mTERI, and then use PCR to amplify C99, and then use Gibson to combine them together.
Oh, okay. I see. I see what you mean. Yeah, I was confused as if I thought you were producing enough plasmids to be like usable. But yeah, never mind. That's a different topic. Sorry. Anyway, good luck.
All right. Thank you.
Allison, I think it's interesting that you brought up the immune effects, because I know that like doing gene therapies and stuff like that, a huge concern is like how the host will react to like the whatever missing gene or protein that they have being put back in. So I know that I think there was one study done like a long time ago, back when like this type of stuff, like this research was really new, where a patient was given he was given the vector for a gene that he lacked, but it had such a bad effect on him that he was only 18 and all of his organ systems failed because of it. And after that, that's really like where the ethics of like doing research like this, especially with like stem cells and like just gene products and stuff like that, that's when it really started becoming regulated. But that's just a tangent. You did a really good job, Mariam.
All right. I guess I can stop sharing.
Oh, and your poster looks really nice too, by the way.
All right. Thanks, guys.
Okay, and I think Renyta and if you guys don't mind, just look out and you should see a co-host thing coming up. We're trying to figure that out right now since we were having trouble with Mariam. But I think you guys should be able to share your screen right now if you guys don't mind presenting.
I'm not able to share my screen yet.
Okay, give us just 1 second. We're trying to make you co-host right now. Is it working now?
Check if you can share your screen now. I think you should.
Yeah, I can do it now.
All right, cool.
So I'm Renyta, and I'm a senior at NC State, and I'm majoring in biological sciences with a minor in microbiome.
I'm Shreena, and I'm also a rising senior, and my major is human bio with minors in forensic science and microbiome.
So obesity is this, it's pretty dangerous disease. It's the condition of having too much body fat, but it shouldn't be confused with being overweight. And as shown in the picture in the bottom left here, you can see that the range for being overweight is lower than the range for being obese. So basically what makes this disease so deadly is that it actually increases your risk for developing other more serious diseases such as diabetes, cardiovascular diseases, and certain types of cancer. According to the World Health Organization, at least 2.8 million people die each year as a result of being obese. And this statistic here on the bottom right was from 2014, and it just shows the significance of obesity, not only worldwide, but especially more so in the United States.
Alcoholism can be considered as a complex disorder here, especially in United States, which can be influenced by genetic factors as well as environmental factors. And it is of importance because researchers have suggested that genetic variation can be one of the reasons for developing alcohol use disorders. And there has been strong relationships between alcoholism with the GABA2 gene, and that's what we are gonna present in this whole presentation.
So genes are complex, and it's not like we were just talking about before. It's hard to just identify one gene for one disease and then proceed to try to knock that out or fix that. Instead, these 5 studies that we've combined for this presentation, they studied something called single nucleotide polymorphisms, or SNPs. And as the name suggests, they are mutations or changes in the genome with just a single nucleotide. Like in the picture shown here, there are 3 sequences that are exactly the same except for one nucleotide. And a lot of times this does not make any major changes, but sometimes there are phenotypic changes or correlations with different diseases. So these studies basically observe the relationship between alcoholism and then the SNPs of the GABRA2 gene and obesity with the SNPs of the GABRA2 gene. And the reason they chose this GABRA2 gene is because it codes for GABA receptors, and GABA receptors are inhibitory neurotransmitters, and alcohol is believed to mimic their effect. And certain GABA receptors are also they're in charge of body weight regulation and satiation, so that's how they are connected with obesity. So researchers try to find this gene's correlation between obesity and alcoholism using biological markers that they placed on the SNPs.
So for methods, because we combined 5 different researchers, they had a basic ground for investigating gene expression and polymorphic effects. For alcoholism, they observed alcohol levels through blood tests, and the subjects for which alcohol dependence was measured, they were measured for both alcohol dependence and their food cravings, just because we are also looking for obesity.
So the studies did find that the gene itself, the GABRA2 gene, correlated with both alcoholism and obesity. However, the SNPs related to this gene were different in the 2 of them. And so they observed that problematic alcohol use increased over time for people with this GABRA2 gene. And they saw likewise the same results for obesity. And for phenotypic results, they observed that patients with alcohol dependence had a significant increased craving for food. So it's possible to interpret these results like GABRA2 gene has some influence on alcoholism, and that alcoholism has some influence on obesity, and that's how the three of them might be connected. But again, correlation is not causation, so we can't say for sure. But this correlation has been observed.
Why is it important to study the relation between alcohol and obesity is mostly for prevention, because as we already saw the statistics that obesity is also considered a disorder as well as alcoholism, and for its prevention we do need to look at the underlying genetic factor which we can find through this GABRA2 gene and how it links obesity and alcoholism. GABRA2 gene also represents an early risk factor for developing both alcoholism and drug use, which is why, again, it's important to look at the correlation between that. And understanding the role of GABRA2 gene as a risk factor for substance use disorder warrants us for prospective prevention methods. So learning how everything works will help us better to have a perspective on different methods that can be used to prevent alcoholism and obesity in at least the United States. Any questions?
I have a quick question, and you might not know the answer. In all these obesity studies, particularly the ones in the United States, do you know if they did any statistics about the obese people and their, I guess they're having that particular allele, like the bad GABRA2 allele, or whichever one's more associated with obesity or not?
Yeah, they did, and they did find that the GABRA2 gene was correlated with obesity, but the S&Ps were different between obesity and alcoholism. So that's what they found. And
you know if the study used any measure for obesity other than BMI?
No, they just looked at body mass index to classify people as obese or not, or at the verge of being obese, or how to distinguish them.
Do you think that sort of like the inherent issues with obesity where shorter people are less likely to make it to obese because of the mathematical calculation and taller people are more likely to classify as obese? Do you think that had any role in how the study played out?
it's possible, but the way that like the body mass index takes height into account, so I don't think it's super biased against any sort of
There's
height.
been a lot of research studies actually done into how it does height squared and then weight, and that like correlation isn't accurate for like obesity measurement in the modern day. It was like made in the 1900s, I think, as a simplified mathematical way to determine someone's height and weight from just one like variable. But there's a lot of like articles out now that like BMI isn't an accurate measure of obesity, or is a not an inaccurate measure, but a much less accurate measure than what we can do today to see how obese a person is.
Yeah, I mean, it's possible because between the 5 studies that we observed, there was like certain studies defined obesity as a little different. Some said it's, above 30 BMI or, above 40. So they do have little differences, but each one just took a number and they stuck to it. So according to that, they found a correlation, but you're right, that could have played a role in the results.
I have another question. Do you know how, like, what the population of the study was, like the demographics and like the age group as well?
We combined 5 different studies, so we did have the population in account, but because we were looking mostly at genetics, we just took the methods and their results into consideration. We didn't really go over all the population statistics.
Okay, thank you.
Were you guys able to find out like why alcoholism like specifically causes memory loss? Because I know that if someone like stops drinking alcohol after like years of abusing alcohol, that it's like linked to like a vitamin B deficiency that also leads to like really severe memory loss. So did any of the studies like you looked at like mention that as being linked to like certain genes the way that this gene was linked to obesity?
So it wasn't, we did see that alcoholism could lead to memory loss, but there was no, they weren't looking at the genes to identify that. They were only looking at, does this gene correlate with problematic alcohol use in general, whether it leads to dementia or whatever, and also obesity. So, but that's interesting.
Any other questions?
Okay. Well, thank you guys. You guys did really good. It was a really in-depth presentation. Thank you. So last week we were supposed to hear from Huma, but Huma, I don't know if you want to present this week or if it's putting you on the spot, but just let us know if you want to go now. And if you do, I think you should have the option to share your screen, but if not, that's okay.
Yeah, I'm definitely completely okay with presenting right now. I'm just trying to figure out if I can open. Yeah, I just got it. Let's see. Is there a way to share like the screen, but like have the like the bottom, like the notes show up for me?
I'm not sure. I think there should be a way to do that.
What was
I
it?
think
I
you
don't know
just
if your notes are like in a different thing, but I know when you're like sharing it, like you can have it so it's just sharing like one thing, if that makes
Like
sense.
one tab, and
Yeah,
then
exactly.
everything else doesn't show up.
Yeah, the notes are just like on like underneath, like on the slide, like the underneath the slides.
I don't know about that.
I think those should be fine. I also have like separate notes that I wrote. So can you share that one more time? Because okay, yeah, actually I think I got it. Yeah. Wait. Okay, well, can you guys see the slide?
Yeah, we can see the slides.
All right. So I did my study on it's a prospective study on leptospirosis transmission in urban slum communities, and it's basically measuring repeated transmission, especially in urban tropical environments. And this study is I think this was a really interesting study because I've done multiple different studies about disease and disease transmission, but this is a cross-sectional study that measures in households and not necessarily individuals, and it takes into account the environment that the participants are in. So just as a background, so it had about 2,000 people in a slum community in the city of Salvador, Brazil. There was a baseline of like 1 year serosurvey, and then there were multiple follow-up surveys for up to 2 years. The measure rate, it was measured through seroconversion, which is just like the specific time. It's up to 4 years that antibody developments can be measured. And then the fourfold rise in microscopic agglutinin titlers are just like they're just like the tells, like that's how you measure if someone has had an inflammation reaction. So in total, at the very end of the survey, like after the first year, there were about 51 participants who did the follow-up procedure, and there was a pretty high infection rate of like almost 38%, 38 per 1,000, excuse me. And the secondary infection rate was higher. So over time, we saw that even though the primary infection wasn't very high, it almost doubled. It more than doubled over time. So let's talk about the structure. So, well, I think I messed up with some of that. So okay, so leptospirosis is just a bacterial disease that used to be found in rural environments, and it was sporadic. It was endemic to where it usually used to be. However, with the rise of urban slums and like just urban populations being very close together and demographic shifts, we have urban hotspots all across I would say like the global south, especially in middle and lower income countries. It's caused by the spirogyety from the genus Leptospira, and it colonizes the kidney of a lot of mammals. And in humans, it can be very asymptomatic, or it could be fatal. Okay. Oh, one more thing I just wanted to mention before. So infection is through either an infected animal reservoir, water, soil, or just any sort of contact with urine. Because of poor sanitation in slum communities specifically, some communities are exposed to animal-borne and water-acquired diseases. So this study was a cross-sectional and case control study, and I think that's really important to mention because there haven't been many slum community-based studies in the past. They're usually done in really controlled environments that don't really take into account all of the additional environmental risks. So this was done in this particular community because it's very secluded, and there aren't very many like ways to have other people get it. So it mostly whatever happens in the slums stays in the slums. The subjects were like, they have poor infrastructure, however, most people who were in this study could this is a long-term cohort follow-up study, so they're going to be followed over like real time, and the data that they produce can be used in a productive manner. So the cross-sectional part just means that they take into account who give me just 1 second. They take into account who the people are that aren't getting any sort of infection, and especially after the primary infection rate, like they keep following up with everybody regardless of what happens with them. The primary host of leptospirosis is the domestic rat. The risk is heavier in tropical urban environments because of seasonal outbreaks such as monsoons, and also there are increased outbreaks because of heavy rainfall and the sanitation issues that creates, especially with open sewers, because of the water that just goes everywhere, and this infections just go wet, and they just go very, very high. And this is just like a quote from like an article that quoted this study in starting to improve sanitation in Brazil. This was used, this study has been used, I think like once or twice in citations by the Brazilian government to actually improve conditions. It's been done with a lot of community action, but I think they're actually making progress on like public health issues because of studies like this. And here's a chart about age-specific attack rates. So looking at this, this is an attack rate chart of overall leptospirosis transmission according to gender. Based on this, we can see that in the 15 to 24 range, we have a lot more transmission over both one second. Yeah, we can see that we have a lot more transmission in the youth population, and also we can see that we in even the secondary population we have a much higher infection rate, but this in the secondary population it increases to 25 to 34, which is a more significant pattern. Okay, yeah, here's the slide about the study site. So the reason that it was done in this area was because the slums of de Lima and like just like that area of Brazil, it's very like concentrated, and it's a smaller area with a large number of participants. Through the study, we also found that the population was basically like the gender divide was basically even, so there was an equal amount of like male and female participants, so we could also measure that as well. So attack rates were measured at 95 interval levels using multinomial logistical regression. This was better to do than using another regression style because it could we could use them. Wait, I'm sorry, my notes are just glitching. Okay, there we go. So yeah, this was better to do because it showed them that like by using a higher odds ratio, we could measure for age and gender, and also proximity to open sewers, and like the socioeconomic status. So this is table 2. It was just in the study to measure like by age, household characteristics, exposures, and also workplace exposures. We can see that in the age 15 and greater than, like to 15 to 34 and greater than 35, we can see that there is a higher correlation between them and leptospirosis. Interestingly, there's no significant difference between the household income and the per capita household income, but the flooding changes a lot. So it's more so like it's like structural. This shows that we don't have a main issue with like even money. If we can't change the infrastructure, like in a lot of other studies, we see that like people come in and they will give like one time like aid, and they will give multiple like treatments. However, if we can see that there's been a 4-time infection rate in the paper, it mentioned that there were people who were having infections not once or twice, but like up to 4 or 5 times. And the study so far, this goes up to May 2014. So it started like 4 years-ish before. I haven't seen like the updated version, but it mentioned that people still had the anti-leptospirosis antibodies up to like this 4 years later, and the percentage was about 25 to 30%. So it shows that even if people are getting better, they're not staying better. So basically, they have a 0.3% chance of getting it again and again every year. Reinfection is frequent, and it's not something that can be solved unless we actually change the living environments that they're in. So effective prevention can't just be focused on short-term treatments and people coming in. It has to be working with the government and working with improving the living conditions. So it doesn't really help to not address the infrastructural deficiencies. This is just like an overview. So so if you are male and you have low income within the community, it increases risk of infection, and it's just more common in young adults and people who live nearest the sewers because of how often they travel outside. And yeah, that's basically it. If anyone has any questions.
Huma, that's really interesting, especially like the last few points that you made, because I was reading an article this morning that said that just having a low socioeconomic status, like at all at any point in life, it will, like, no matter what your socioeconomic status is when you're older, the fact that you even had one when you were younger, you're more likely to mirror like stats for that group's health later in life than you are for whatever your current like socioeconomic status is. So it's really crazy, like how even just having a low socioeconomic status, like in childhood, can like stay with you for your entire life. Like I feel like that's something that's not really talked about a lot, and it's really, I think it's really scary. Like
Yeah, and I wanted to mention some more of the infection rates, but it's just like a lot of data. So I thought it would be better just to present like the words for it, I guess. But yeah, it's very much like it doesn't really help to like have the same models for different communities unless you address like what they're going through. So yeah, like so like even like Charlotte, we can see that like communities who are poor will stay poor and they'll have the same sort of health effects. But yeah, that study sounds really interesting as well.
I couldn't tell, but did it say did it show if males or females had higher rates of infection? Like was that in one of those graphs?
Yeah, let me I can reshare it. There was like yeah, in table, I think it was like table 1. Yeah, figure 1. There's an age specific and also gender specific. Yeah, let me just reshare this page. Yeah, so basically if you're a male, you're more likely to, I think like it's just like going outside more, and it might be contributing. Like I don't, obviously causation is not correlation, but we can see here like with male gender, there's like just so much more, like even with like the odds ratio and everything, like it's just so much higher. And interestingly, there's nothing like it doesn't correlate if you're like not as literate. It's just like being male and being poor, and living proximity, like the proximity is closer to the sewage system. So like that's all. Like unless you can move, it doesn't really help.
Yeah, do you think it's just based on like I don't know, like traditional male jobs, putting them out like in more, like more exposed environments or something like that?
I think so. I wish I had more context about the study because I looked it up, like, but I couldn't really tell because like there's just not that much information in English at least. But yeah, I think that might be it, like from what I know currently.
But the thing is that, like, even in the US, like, like male like health versus women's health, like women live longer and stuff like that. So it's probably jobs, but like it's also like honestly men just don't take care of themselves as well.
Yeah, I agree.
And I guess that's an international thing.
There's also like a big cultural thing to think about in research between gender. So like it could easily be like they just don't report on women as much as they do men, because there's a lot of issues in medical based research where women aren't including in research studies, and those who are LGBTQ plus who aren't included in research studies. So there could be that imbalance.
Good point.
Yeah, that's definitely true. Oh, sorry. You go ahead.
Go ahead.
Yeah, that's definitely true because lots of studies that use animal models only pick the male to do all their studies on, which is not representative of like female populations, but they do it because like of this like old way of thinking that, females have these additional like hormones and these additional factors that they have to contend with. So like, I mean, I don't know if they do that in human studies because
they do.
That's very sad and unfortunate, but
yeah.
I think.
Sorry.
Yep. I just think that also in rural areas, you have to account for the fact that a lot of women aren't educated, right? Like only the men are educated, if even that. So it's going to be hard for someone to come from like America or a first world country and get accurate information from women who aren't educated at all, or aren't like, they can't even read in their own language, write in their own language, much less speak English or write in English. Right? So it's like, even for my grandma, like it's hard for her to like I talked about this before, but it's hard for her to describe her pain to like American doctors and stuff like that. Same thing with my grandpa, right? So like if you have like other people going to third world countries who don't speak their language, it's hard for them to get an accurate description of like what they're going through, I just think.
Yeah, those points are really valid, especially about like the pain scale. Like there's been a lot of well, not studies, but there's been a lot of discussions about like how it doesn't really work because people measure pain differently. Like if you're in chronic pain, you're going to like something that's a 2 for you might be an 8 for someone else. But yeah, like about the outsiders coming in and just not having ideas about how to communicate. So the study also did show that women actually between the ages of 15 and 34 had an increased level of secondary transmission. However, it was a lot less significant than men. So I think like earlier, like we were talking about like occupation. I think that might be a big part of it, but also like in this particular study, they partnered with like Brazilian like hospitals and also like Brazilian like clinics and volunteer workers. So it wasn't as I guess like it like they weren't just coming in without any like cultural background. They had some cultural context and they were doing households. So it wasn't just one person. So when they measured the transmission rates, they would go directly to their house and they would have people come from the clinic to do so. Like, so they had translators and they also had like, and I don't remember exactly where in the study they said this, but they had like female practitioners for the women so they could feel more comfortable.
Oh,
And
that's
like,
actually really interesting.
Something I wanted to ask, does the study talk about how much it would cost to like go into a Brazilian favela or like any like, you know, a poor town in a third world country, update the infrastructure and then maintain that infrastructure going forward?
So it talks about the UN habitat, like it talks about like some of like how to do that, but basically they say that unless they get funding for it, they have no power to do so. They basically just wanted to create more data for like some communities. For example, like in like Mumbai in 2005, there was a huge outbreak of I think it was leptospirosis as well. Like, and there was nothing to like help because nobody was going in previously. So yeah, no, it doesn't really say a lot of that stuff. Like I would have loved that information, but I don't really think that like they have that information either. They also say that like there's a lot of limitations and they like the crude rate didn't necessarily what is it called, the crude rate and the adjusted rates weren't necessarily the same, and like there's a 21% like prime, there was an out migration from the study, so like 21% people who started the study never finished. So like a lot of them were men, like a significant portion of them were men, and a lot of them were better educated and had like a lot, like I guess like a lot better like life outcome. So this study, while definitely really important, isn't conclusive. So yeah, I hope that kind of answers the question. Yeah, but the original information that you asked for is just not there.
I was gonna say, because I've actually I've done some reading on a lot of corporations to like for recompensation, they'll go into third world countries and try and set up like social support programs or, you know, sanitation initiatives and stuff. But one big problem that occurs in a lot of those initiatives is they'll bring in American workers to maintain those initiatives for like the year and a half that they actually fund them. And without training or educating the actual populace to maintain the initiative once you're gone, it falls into disrepair in like a year and a half at most. And no one's going to come in and repair it once you're gone. So, and I was just wondering if there was like any sort of economic outlook on like how you would actually effectively go into a third world country and try and address the sanitation issue.
Yeah, that reminds me a lot of like whenever there was like the Ebola outbreak, like back when I was in high school, so I think a couple of years ago, how all those documentaries that were being made about it and stuff, it was all centered around the issue of like, okay, we have all of these healthcare workers like from like the Western world coming into Africa, but a lot of them are not trained to be culturally sensitive or how to really communicate with like local populations and stuff like that, like in a way that like takes into account like their cultural practices and stuff like that to like maintain better health. So that just kind of like reminds me of that.
And I think that's something that can be applied in just about anything, right? Like, it's one thing to have like some person from like halfway across the world telling you to do something. It's different to have somebody else that speaks the same language as you telling you to tell you what to do, though, if that makes sense.
And it's also, in my opinion, it's always been easier for me to do something when someone explains why I'm supposed to do it. Like, I always hated when my teachers gave me busy work and were like, do it because I said so. But then if someone explained, like, doing this will teach you how to apply this in X scenario, I would become much more interested in the work, because now you know that it's like usable. It's not just some random guy telling you, hey, you have to do this because I said so, because I have some authority over you. That's, you know.
Yeah, and unfortunately, like a lot of this, like it says that a lot of the study, it kind of just says that they're at the mercy of like the organizations that sponsor them. So like these researchers, like though they have like PhDs and more information, they can't really do anything with that unless the Brazilian government approves, unless like, because it's in partnership with like Yale and other like, you know, like big like colleges and universities. So like until they can get that like the type of support from not just like these organizations, but from like the people who live there, they can't do anything. I read, this isn't like in the study, but like I read about the study, and they're doing something similar where they're training the doctors like more about slum communities and how to address that. And similar initiatives have been taken in Mumbai, especially after that 2005 outbreak. I think it started in 2011. But yeah. Is there anyone else who's okay? Yeah, it's already 2 o'clock, so no one else is any. Yeah, well, that's everything.
Well, if no one has any more questions, we'll end it here for today. And I think there's a lot of new people who don't come regularly, so I just want to mention that next week is our final meeting for the summer, and we will continue this club for the fall, and hopefully, you know, it'll stay a club at state for a long time to come. So come next week, because we'll be talking about how we're going to go about doing stuff in the fall, any new officer positions, and a t-shirt contest. So I hope to see everyone there next week. And thanks everyone for presenting. They were all really good. Bye guys.
Thanks.